Good afternoon to everybody. Welcome to the 16th seminar of the Bridges Seminar Series, which is organized by the Hellenic Initiative Against Alzheimer's Disease. The aim of this seminar series is to focus on topics that promote the dialogue, the discussion, the interactions between basic scientists and clinicians. The speaker of today's seminar is Milap Nowrangi, and he will talk about the biological basis of neuropsychiatric symptoms of dementia. Our speaker is an assistant professor of psychiatry and behavioral sciences at the Johns Hopkins University School of Medicine. Dr. Nowrangi specializes in the diagnosis and treatment of age-related cognitive disorders, including Alzheimer's disease and other dementias. He is also interested in the treatment or management of behavioral and cognitive symptoms of neurological diseases, including those caused by traumatic brain injury, stroke,
Thank you, Dr. Alexopoulos, and everyone here for joining. I made the comment earlier that this is the closest I've gotten to Greece. However, we had dreams of coming there this summer. I was instructed, however, by several people to avoid the summer, so it'll have to be some other time. But I thank you all for joining at 5 o'clock your time. I know it's early evening, and you'll more enjoy the time after the talk when you can go visit your closest beach for a beautiful sunset. I'm sure it never gets old there. So I wish I was there, but I'm here nonetheless in equally beautiful Baltimore. But I would want to thank the organizers of the seminar, as well as you, Dr. Alexopoulos, and Dr. Soteropoulos for the invitation, and for Dr. Laketsos for making the connection. And I'm happy then to present this talk sort of a high level. Let me share my screen. I hope you can all see the screen. Okay. So I'm happy to present this this next 40 or 50 minutes or so. It's basically an attempt to review at a fairly high level neuropsychiatric symptoms, the etiology and the neurobiology of them, in the context of a new framework that we're calling an anatomic genetic framework for developing eventually better treatments for these unfortunate symptoms. And while I'll say this at the outset, is that while the framework includes consideration of both anatomy and genetics, this talk will primarily focus on the anatomic, in large part because our group here with Dr. Laketsos, my colleague Paul Rosenberg, and others have done more work with neuroimaging than with genetics. But we feel that it's an exciting time to move also to genetics in the future with other collaborators. Some may be at your institutions. Okay, so as an outline, I'd like to suggest that we start with a fairly heavy dose of theory, starting with a clinically grounded case, then discussing phenomenology, making a case for a disconnection as a way to understand neuropsychiatric symptoms, and then an embarrassingly small amount of the talk will be given towards genetics. And then finally, we'll review what we've discovered here with the paper that we published last year, suggesting that there's an anatomic and genetic framework for understanding these symptoms. I will also add that I have no disclosures. First, I'd like to tell you a little bit about my dear late mother-in-law. She was a retired pediatrician trained in India, where we're from, and practiced in Southern California for the last 40 years. About 10 years ago, she sadly developed cognitive deficits, primarily around memory and decision-making, and then went on to develop worsening of these symptoms, but then with the added difficulty of experiencing changes to her personality and behavior. Early on, we noticed that she became quite passive, reserved, didn't want to get up out of the chair, but perfectly content, staying at home, drinking her chai at least once to 3 times a day, And that was really the most of her motivation was to do that. One other thing that you could potentially know about her is that when she was in her 20s, she lost her firstborn son in an awful auto accident in India, and then went through some period of undisclosed psychiatric treatment earlier in her life at that age. And of course, we never discussed that. But it was clear to me anyway that she had, at the very least, residual symptoms of post-traumatic stress disorder and depression, having periods of low mood, anxiousness, and fretfulness, being avoidant of certain situations. She often grabbed my wife's arm when crossing the street, even though there were no cars around. And she should also know that she was very hard of hearing and suffered from chronic ear infections as a child and was essentially deaf with little improvement from hearing aids near the end of her life. As her cognitive symptoms deepened over the succeeding years, she developed even more problems with emotion and behavior, and then developed, unfortunately, we believe new onset seizures. She might have had them before in her life. But with this, she was hospitalized one time after a fall with mental status changes and was found to be in status epilepticus. With stabilization, we also found that she had fractured a hip. She was repaired and then stabilized, and then came home with, unfortunately, a profoundly altered cognition and changes in her personality. She was once docile and sweet, but then developed periods of irritability, obstinance. She was demeaning. And we noticed that she had also developed paranoid delusions, believing people were stealing from her, claiming that she was in the house with somebody else when no other people were there, and there were bugs crawling in her food. All this in the absence of being necessarily delirious. As all her symptoms developed and continued to worsen, she sustained another epileptic seizure, unfortunately, this time which was intractable and one from which she didn't recover. I tell you this story because of obviously my firsthand experience with these symptoms. I lived it firsthand. My sister-in-law in California lived it firsthand. She was right by her side. And what we learned was that she likely had a mixed dementia where her brain imaging showed severe atrophy globally, as well as vascular brain disease, several areas of ischemia. And I mentioned this also as a personal story to set the stage for this discussion, which I hope we'll have. But first, I think it's useful that we ask ourselves, as most talks on neuropsychiatric symptoms go, to understand what the problems are with these neuropsychiatric symptoms in Alzheimer's. First, we know that Alzheimer's is more than just cognitive symptoms. In fact, they include the mood and behavioral symptoms that we're about to talk about, as well as functional decline, an area that I spend a lot of my time is understanding how individuals develop complex decision-making problems, particularly around finances, and that all these together lead, unfortunately, to loss of independence. These behavioral symptoms are nearly universal, with most common ones being agitation, apathy, and depression. And until recently, there are no approved drugs, quote unquote, approved drugs for the symptoms that we're discussing. And the ones that are used are often used off-label and are repurposed drugs originally developed for the treatment of idiopathic psychiatric diseases. And when using some of these drugs, we have found through practical experience and through studies that they have limited effectiveness and at times even no effectiveness. The symptoms lead to negative outcomes that I'm sure we're all aware of: accelerated cognitive decline, institutionalization are just a couple of those examples. Of course, caregiver burden is one we see all the time in the clinic. And ultimately, none of the drugs that we use target the causal mechanisms where it has been hypothesized that there are both direct and indirect pathways that lead to these symptoms stemming from the brain changes of Alzheimer's disease. So ultimately, the observation that some treatments only work some of the time begs the need for a better understanding of the mechanisms. So let's now take a little step back and engage in a little bit of theoretical talk with a brief discussion of the phenomenology of neuropsychiatric symptoms to set the stage then for a wider discussion on brain structure and function. So when I teach the fellows here, I lead the Neuropsychiatry Behavioral Neurology Fellowship here. I tell them that our understanding of how these symptoms develop and how we talk about them follows inductive reasoning. This is a logic where similarities between two ideas are made and sometimes, unfortunately, conflated and used interchangeably. This is the whole, if it walks like a duck, talks like a duck, it must be a duck adage, where in this case, the neuropsychiatric symptoms appear to be closest to those exhibited by individuals suffering from idiopathic psychiatric conditions, ones like bipolar disorder, schizophrenia, or major depressive disorder. So, for example, individuals suffering from lack of emotional reactivity, low mood, poor appetite, and disordered sleep appear to be depressed in the way that we understand major depression, and so we call it depression in Alzheimer's disease. Or if an individual exhibits disordered thought content, is observed talking to themselves, or has a disruption in another modality of perception, these can be thought of as suffering from psychosis, like in schizophrenia, and so we talk about psychosis in Alzheimer's disease. And so while this method is effective because we can accept a commonly agreed upon terminology and taxonomy, it is helpful in forming hypotheses, but it's not as good as applying and testing the hypothesis because it is unclear if the observed symptoms are in fact the same. So we need better tools to tell the difference between them, and the better ones, quote unquote, better ones that have been developed have been ones like the neuropsychiatric inventory, which, as you know, is an informant-based interview to assess neuropsychiatric symptoms over the previous month. It involves 10 neuropsychiatric domains and records the severity of the symptoms to the patient and distress to the caregiver. And as you can see, the terminology used here is not unlike that used in general psychiatry as put forth by the DSM. So it turns out that this isn't the only way of describing pathological human behavior. Our professional twin siblings, the neurologist, we were separated at birth, raised in different countries, employ a different vernacular, right, to map better onto the brain structure and function. Some people have called this so-called neurobehavior. And within the constructs, we find that observation of pathological human behavior are considered in relation to large brain structures such as the frontal and prefrontal cortex, which give rise to disorders of executive function and individual comportment, how one conducts themselves. For example, a syndrome of frontal disinhibition is described as an individual who displays traits that are puerile, profane, slovenly sometimes, facetious, and grandiose, symptoms consistent with damage to the orbitofrontal and medial frontal cortex. A syndrome of frontal abulia, in which the individual will have lost spontaneity and curiosity, and initiative and drive, as well as apathetic blunting, we call that also affective blunting, are symptoms often seen in damage to the dorsolateral frontal cortex. And finally, syndrome characterized by loss of insight, judgment, and exhibiting concrete or stimulus-bound thinking are symptoms that we often see in damage to the prefrontal regions. And then there are structures that are involved in the linkage of cognition with the hypothalamic states. These are the states such as temperature, heart rate, and hunger, and those linked with emotion. These are the functions of the limbic and paralimbic regions. So memory, as part of the paralimbic and limbic regions, we'll leave mostly for now, but we know it is well related to the hippocampal related centers and have redundant and reciprocal connections to structures like the amygdala and the anterior cingulate, the ACC, which plays pivotal roles in the expression of emotion and affiliative behavior. In fact, the amygdala is central and critical to channel drive and emotion, where people like Marcel Meslim have identified the structure as being essential for having a stable mental state. A stable mental state is dependent on a relatively predictable interaction between experience and emotion, so that pleasurable experiences and thoughts lead to positive emotions. The extreme example of damage to the amygdala and surrounding structures is in a syndrome that you may know of called Kluver-Bucy syndrome, which is a widespread damage of the anterior temporal lobe, including the amygdala. And at least in primates, we know these individuals have indiscriminate sexual activity, oftentimes with non-sexual objects. They lose the aggressive and aversive response to others, and they engage in hyperorality. The other function of the amygdala is adding emotional valence to some sensory stimulus, right? That is characterized by an innate type and degree of pleasure, a characteristic held by that object. So for example, it is joyful for me to see my children, at least most of the time. And so I would run up and give them a hug. That's how I interact with the environment that my children occupy. And the internal emotion that I experience is towards that innate characteristic of my child through various sub-mechanisms. And so secondly, these regions, specifically the amygdala, the hippocampus, anterior cingulate, orbitofrontal, and temporal pole, are also responsible for linking visceral states, such as heart rate, blood pressure, GI motility, salivation even, and sweating, right? Which is likely why visceral experiences are so common in conditions like temporal lobe epilepsy. In the case of sitting down to a delicious meal, for example, there's typically an increase in my heart rate, I salivate, my stomach starts to grumble, and so on. Unfortunately, in maladaptive states where they're such as under chronic stress, those same positive emotions and those same visceral experiences may lead to excessive drive and create things like GI ulcers, cardiac arrhythmias, and things of that nature. So what we also know is regardless of which taxonomy we use, one from our twin, the psychiatrist, and the other from the other twin, the neurologist, we know that these symptoms tend to run together in clusters. Using latent class analysis of 198 Alzheimer's patients and 5,000 older adults in the Cache County study, Dr. Leketsos is the expert here with that. Individuals with primarily affective symptoms comprised about 30% of the group, while those with psychotic symptoms, 19%, and another group that they called monosymptomatic disturbances, another 19% of the group. Another study identified another cluster that they called hypomanic symptoms, and yet another study by Cheng et al. Used factor analysis and found 4 clusters. One cluster around what they term behavioral problems for symptoms such as agitation and aggression, disinhibition, irritability, and aberrant motor behavior. One cluster for mood disturbance to describe depression, apathy, and sleep, and appetite disruption and psychosis. The third cluster being psychosis, those involving hallucinations and delusions, and finally a cluster that they termed euphoric symptoms. So I'm not sure ever if I developed this on my own, in my own thinking, or if I read it somewhere. It all sort of kind of runs together after a while. But my observation and experience from my clinical work gives rise, in my mind, at least to 3 different types or categories of symptoms. One are symptoms involving disorder of emotion and affect. That is, the affect being the outward expression of the internal experience of emotion. One where you experience or observe depression-like symptoms in Alzheimer's, which are reflected in states of low mood, anhedonia, restrictive affective blunting, psychomotor retardation, for example. And secondly, an apathetic or abulic subtype, where it consists largely of loss of initiative and drive, complacency, excessive deference, things like my mother-in-law exhibited early in the course of her disease. And then secondly, a grouping around psychotic symptoms or disruptions to perception or belief. These are the thought contents, where symptoms such as internal preoccupation, somatosensory disturbances in all 5 sensory modalities, hallucinations, that is, and disorders of belief or thought content, such as paranoia, imposter delusions, somatic delusions, which are very common in this population. And finally, a grouping of disrupted conduct. These are the control of one's personal behavior and comportment, where common symptoms are things like asocial symptoms such as disrobing, verbal disdecorum, asocial behavior, impulsivity, and physical aggression. So as painful as that might have been to go through in that level of detail, and the theory behind why we have behavior, how that is reflected in normal as well as abnormal states, I think it's important to talk about that in the outset, if for any other reason than to illustrate how complex, messy, inexact it is to describe the human internal state, specifically as it relates to emotion and drive, and its relation to how we perceive and interact with our external environment, broadly considered behavior, human behavior. And as such, I think it's really no wonder that we struggle to have effective treatments. We can't even really understand the experience, the observations in the same way. And if you think about not being able to speak the same language, it's really hard to come up with effective treatments for it. So we need a more precise construct and taxonomy to understand these symptoms. In an effort then to become more precise, I think we need to identify the etiopathology of these symptoms. One very simple model, there are many, but one simple model involves understanding that genetic expression gives rise to brain structure, yes, but also function, in part with neurotransmission, right? Either chemical, electrical, metabolic, energetic, what have you. And then the structure and function is affected by disease processes, resulting in disruption to the larger scale functional circuitry that the structure of the brain creates, right? And that this altered functional circuitry then gives rise to these clinical symptoms. In recent history, over the last 20 years or so, the study of neuropsychiatric symptoms has gained a lot of knowledge from neuroimaging that has given rise to hypotheses of these symptoms as disconnection syndromes. We'll talk about those in a minute. But disconnection of these coherent neural networks. And I think that that's particularly our strength here, is to understand those networks in terms of brain imaging. And so we'll go into that in some detail. While also there have been advances in genomic sciences where have been identified specific gene associations to discrete symptom phenotypes. So since brain structure then is an intermediate phenotype of genetic expression, integrating results that we have had from neuroimaging and genetic research on neuropsychiatric symptoms of Alzheimer's disease in the same framework seems like a reasonable goal. So I hope I won't take up too much time here, but I thought it would be very good for us to understand what we mean by disconnection and how that relates to mechanism in neuropsychiatry. So I thought I'd make a case then for this being a potential mechanism that we ought to understand well. As a historical aside, the idea of disconnection came from a number of luminaries in the field of behavioral neurology, people like Theodore Meinert, who detailed the anatomy of white matter pathways and described projection fibers, both ascending and descending, commissural fibers connecting the two hemispheres, and association fibers of cortical regions within the same hemisphere. Carl Wernicke, who described conduction aphasia with the arcuate fasciculus, often considered the father of disconnection theory. And then Norman Geschwind, the father of behavioral neurology, who described a disconnectionist framework that revolutionized the neurosciences, and his two progeny who are still alive, Antonio Damasio and Marcel Meslin at Northwestern. So there are some examples I'll just quickly mention here of well-described disconnection syndromes. One is disconnection to the arcuate fasciculus, which produces a conduction aphasia. The uncinate fasciculus, where damage to that results in verbal memory retrieval and long-term memory problems, also some self-awareness issues. The anterior cingulate cortex, which you'll hear much more about, which has deficits in emotion, akinetic mutism, abulia, and apathy. And of course, the large white matter bundle connecting the two hemispheres of corpus callosum with a variety of conditions that result from its damage. So I think it'll be also useful then to make a case that disconnection is a cause for behavioral disturbance. And while we skip a lot of knowledge here, I think it's important to also say that we must accept that neurological disconnection syndromes are a result of damage to white matter axons and communication pathways between cortical regions that are independent necessarily of cortical damage. And secondly, understanding functional disconnection in terms of behavior requires appreciating that large and small scale organization of the brain, including the function of cortical regions and how they are connected to each other through white matter pathways that run typically together in white matter fiber bundles. And that put together, these give rise to behaviorally relevant networks of cortical regions. Marcel Meslin described 5 large-scale networks of these cortical regions connected together. 1, a dorsal prefrontal network for spatial orientation. 2, a limbic network, as we described earlier, for emotion and memory. 3, a perisylvian network for language. 4, a ventral occipital temporal network for face and object recognition. And 5, an executive function and comportment network that we also introduced earlier. And also as a result of these presuppositions, Jeffrey Cummings and others have described that there are 3 behaviorally relevant circuits, all with origins in the prefrontal cortex: a dorsolateral prefrontal circuit, which mediates executive function; an anterior cingulate circuit, which is involved in motivational mechanisms; an orbitofrontal circuit, which has lateral and medial divisions; and that are common to all these circuits is an origin in the frontal lobes, with projections sequentially to the striatum, to the globus pallidus, and then to specific thalamic nuclei, with the final link back to the frontal lobe. Our group, myself, Dr. Leketsos, Dr. Rosenberg, took an initial drive into trying to understand these circuits by reviewing the neuroimaging literature. It seems almost like yesterday, but this was all the way back in 2015. And what we did was we reviewed the neuroimaging literature and looking for consistent atrophy in brain regions and their associations with neuropsychiatric symptoms. And so we found that atrophy within the frontal cortex, the anterior cingulate, posterior cingulate, amygdala, and hippocampus were highly related to individuals displaying symptoms of agitation, while atrophy in the orbitofrontal cortex, frontal cortex, inferior temporal cortex, ACC, and PCC were found in individuals with apathy. Our group also examined clinical and neuroimaging data collected from the medical records of outpatients. So this is not a research study. This was a real-world study of active clinical patients that were seen in our memory clinics. Myself, Dr. Leketsos, all our other clinician colleagues see these patients. And we gathered their clinical data from their medical chart, including scales of neuropsychiatric function like the NPI, as well as their neuroimaging that was gained on clinical grade scanners. And we processed their images with an MRI analysis that used an automated multi-label atlas fusion method in which brain volumes were measured in a priori determined regions of interest. Regional brain volumes were then correlated with agitation, apathy, and delusions, and were as such carried from a linear regression analysis. So for 72 patients that had clinical and usable neuroimaging data, many of them didn't because of the quality of the scan, we found that agitation was inversely associated with rostral anterior cingulate cortex bilaterally and the left subcallosal ACC volumes in a group further characterized by moderate severity. Delusions were positively associated with the left ACC volume in both severe and mild groups, severe and mild severity groups. And what this research was essentially, boiled to its core was that the anterior cingulate cortex, we believed, had a prominent role as what we call the behavioral node in individuals with cognitive impairment, many with Alzheimer's disease. So I'm going to switch gears for only one slide here, and this is an embarrassingly general and short slide about the behavioral and psychiatric genetics related to Alzheimer's disease. But as I said in the beginning, the focus of the talk was on structure and anatomy, and so I'll ask for your forgiveness again in not doing this topic justice. I might add that a co-author for the paper that I'm about to review that we wrote is Dimitri Avramopoulos, a fellow Greek, and I might humbly suggest that you ask him to give a seminar on the genetics of Alzheimer's disease and these behavioral disturbances. So why should we study genetics in the context of Alzheimer's and neuropsychiatric symptoms? First, we know that genes are heavily involved in Alzheimer's disease. Of course, in mostly early onset Alzheimer's cases, an autosomal dominant form is seen and characterized by mutations on one of the three commonly discussed genes, presenilin-1, presenilin-2, and the amyloid precursor protein gene. That said, even in late onset Alzheimer's disease, it is found that the condition is highly heritable, up to 76% of it. And to date, the ApoE gene, specifically the E4 allele, has been accepted as a genetic risk factor. There's also now additional data in late onset Alzheimer's disease that the SORL1 genetic variations are also associated with, again, late onset Alzheimer's disease, as well as many other genes. But because of genetic heterogeneity of the disease, there have not been confirmed studies that have pointed in one direction towards them. Moreover, treatment of neuropsychiatric symptoms, specifically with psychosis, has been suboptimal due to the limited efficacy of the classes of drugs used and their high toxicity in the group, one reason why we should study genes. And so we need then to have a better and improved idiopathological basis for developing or improving these drugs. And if you remember from the model that I presented earlier, it all starts with our genes and the genotype that we can see from them. Let me also add that the methods used to carry out these studies, these genetic studies, are mostly out of my grasp, but the ones that allow for analysis of large populations, such as genome-wide association studies, GWAS, in which the entire genome is analyzed, sequenced for mutations and SNPs, through looking for using analyses like candidate gene and linkage analyses are the primary methods for understanding how the genome has changes that are related to these clinical symptoms. And so the clinical symptoms on this background, typically as behavioral clusters that we defined earlier, may be recognized, which are likely linked either to the selective loss of specific genes coding for a neurotransmitter, for example, or have other common genetic bases. Among the neuropsychiatric symptoms, major attention by people like Robert Sweet at the University of Pittsburgh has been given to understanding the relationship of the genetic composition of individuals who have dementia and psychotic symptoms. Some authors have proposed that polymorphisms in dopamine and serotonin-related genes may be interesting candidates for the neuropsychiatric manifestations of dementia. And among those genes, particular interest has been given to the catechol O-methyltransferase, the COMT gene, which is involved in dopamine breakdown, and which contains a functional common polymorphism. There's also been evidence that the serotonin activity is regulated by functional polymorphism within the promoter region of the 5-HT transporter gene. So finally, we come then to this systematic review that we conducted and published last year that kind of puts together all this information on the backdrop of the theory, on the backdrop of the clinician who sees a family member with these terrible symptoms. And hopefully what this will do is give us a chance to put it all together and understand the etiology of these conditions so that we can think of better ways of treating these diseases and the symptoms of them. So what we did is we organized the associations found within the neuroimaging studies by cortical brain region, frontal, temporal, parietal, occipital, but we also added the subcortex white matter when we reviewed all these papers and studies. And the associations found in the genetic studies were organized by predominant functional expression of the gene product themselves. This is a model that Michelle Sweet and colleagues at the University of Pittsburgh also modeled in one of their papers, which is dividing up the gene products according to neurodegenerative gene products, inflammatory, vascular, monoamine, for example, and so on. Okay, so what did we find? This is a terribly busy slide. I apologize. I should have sent the paper ahead of time, but after the talk, I'll be happy to furnish all of you with the paper. For depression symptoms in Alzheimer's disease, we found 22 neuroimaging studies of depression symptoms in Alzheimer's, with the largest number, 12, reporting associations with the frontal lobes and the structures within it. The anterior cingulate cortex, in particular, was associated with depression symptoms, and you'll remember from the study we did of clinical patients, the anterior cingulate cortex was brought up there as well. Genetic studies of depression symptoms in Alzheimer's showed variable associations with genes involved in monoamine synthesis and function. One paper, one study by Arlt et al., found a positive association between the intronic SNP related to referred to as A218C in the tryptophan hydroxylase gene TPH1. And as you might know, tryptophan hydroxylase is involved in the first and rate-limiting catalytic step in the biosynthesis of serotonin. For psychotic symptoms, neuroimaging results from this review showed prominent associations within the atrophy of the temporal and parietal lobes, and less so with frontal and occipital lobe structures and subcortical structures. As in depression, increased atrophy of the hippocampus was associated with psychosis. In addition to showing associations within the entire parietal lobe, the posterior cingulate cortex atrophy was also found to be positively associated with psychosis in several of the studies. The literature linking psychosis with genes is robust, with 26 studies showing association with the majority focusing on genes expressing some product of the monoamine synthesis pathway. Genes involved in serotonin synthesis and function were also prominently seen in the literature, with 9 papers showing association with psychosis. And genes coding specifically for dopamine synthesis and function, the dopamine receptors and COMT, for example, were also associated with psychosis, as were genes or gene products involved in the neurodegenerative pathway, genes like the ApoE gene, MAPT, NMDA receptor gene, which are active in the synthesis and function of glutamate, as in the case of NMDA, and tau, as in the case of MAPT gene. So, though neuroimaging studies of these behaviors have been limited in large part because of the practical challenges of scanning agitated and cooperative patients. You can imagine trying to get a agitated patient and to sit still in a scanner for 30 minutes. There have been some associations made between agitation and neuroimaging. One study by HU et al., through a secondary analysis of the ADNI dataset, found a relationship between agitation and atrophy in the left inferior frontal cortex in the insula and the bilateral retrosplenial cortices, using a voxel-based MRI method. Another study using diffusion tensor imaging by Sarah Teig, who was actually a fellow and a co-fellow of mine, found that decreased fractional anisotropy, suggestive of decreased white matter integrity in the anterior cingulum bundle. There's the anterior cingulum again, in mild cognitive impairment in Alzheimer's disease patients, tended to be associated with agitation as well as irritability and apathy. And though the limited evidence base in genetic literature was found, the studies we did found reported associations with genes coding for the monoamine system again, specifically dopamine and serotonin synthesis and neurotransmission. So here is in graphic form are the three neuropsychiatric symptoms we focused on, which are overlaid on brain regions asserting the associations. The size of the shapes of these shapes, the circle, the triangle, the square, reflect the number of papers asserting the relationship. So you see a large number of papers on depression asserting association over the temporal lobe and the frontal cortex, and much smaller ones with, let's say, agitation and the parietal lobe, for example, or the occipital lobe. And then with respect to the genetic literature here in graphical form is a table that shows a number of papers of particular gene function associations with neuropsychiatric symptoms, specifically showing the largest number of papers asserting relationships between monoamine and neurodegenerative genes and psychosis, and relatively smaller with other neuropsychiatric symptoms. So, what do we say in summary? We have then proposed a rational anatomic genetic framework on which to build future treatment development for neuropsychiatric symptoms of Alzheimer's disease. The neuroimaging and genetic literature support brain mechanisms underlying monoaminergic biosynthesis and function, as well as brain regions, ultimately networks, which we haven't got to because we've studied primarily structure, and the genes affected by or involved in neurodegeneration. Specifically, all three neuropsychiatric symptoms that we studied or analyzed demonstrated atrophy in frontal, temporal, and parietal lobes, including the ACC and PCC and hippocampus, all of which are heteromodal association cortices integrating emotion, sensation, and motor responses. And all three symptoms that we studied involved genes that code for monoamine biosynthesis and neurotransmission, especially ones coding for regulatory monoamines, serotonin and dopamine in particular, and those involved in the neurodegenerative pathway, as I mentioned earlier. Therefore, there seems to be a clear connection between anatomical locations of the neuroimaging findings and the neuronal projections of serotonin and dopamine, which are coded by the genes found to be associated. So very quickly here, this is something I use with the fellows, but you can see here that the projections of serotonin across the brain stem from the brain stem in the medial and dorsal raphe and innervate most of the brain, including the cerebellum and spinal cord. Norepinephrine are ascending projections, again from the brainstem, from the locus ceruleus, overlapping serotonergic projections, and as we all know, have an effect on mood, arousal, and cognition. And then the dopamine projections are ascending projections from the brainstem in the ventral tegmental area, the substantia nigra, and are involved in reward cognition and psychosis. So to bring it home, we should take another step back and look at the evidence raised by 20 years of the neuroimaging and genetic literature. And I think we can conclude that the current treatment approaches are oddly in line with what we understand as the mechanistic underpinnings of these symptoms. Okay, we haven't completely bored those out, but the literature seems to suggest, as we talked about earlier, the brain regions underlying the neurochemical changes and the genetics underlying these neurochemical changes should give rise to the medications we commonly use, right? So the psychotropics, the antidepressants, rather the serotonin selective reuptake inhibitors, for example, or the antidopaminergic medications should produce positive outcomes. Why don't they? One reason for this might be that other mechanisms are at play that are outside of neurodegeneration and the monoamine system that we described. As an example, depression involves genes not only for monoamine and neurodegeneration, but also neurovascular function, as well as inflammatory and immunologic cascades that have also been reported in the literature. These other mechanisms have not been well researched, but may prove to be promising targets for future development. Secondly, it should be noted that targeting single mechanisms is not likely to address the complexity inherent in behavioral syndromes in general, because they're so complicated. And certainly the underlying anatomy of these distributed networks, large parts of the brain involved with underlying structure and function, would be an evidence of that, and not completely understood by the neuronal projections of the neurochemicals. So we'd often talk about a cocktail approach for the treatment of cognitive symptoms in Alzheimer's, which has been gaining interest primarily with our investigation in amyloid therapies and also potentially now tau therapies and others. And there have been also talk of including anti-inflammatory and other mechanisms for the treatment of cognitive impairment in Alzheimer's disease. And the question then remains is, because there is such heterogeneity in the clinical symptoms of Alzheimer's, there is an increasing interest in developing a precision medicine approach where the same type of heterogeneity in cognitive symptoms may also exist with neuropsychiatric symptoms. And so one might even conceive of a cocktail approach in treating these neuropsychiatric symptoms. Speaking of the precision medicine approach, I think it's becoming more clear that individualized treatment will benefit from this anatomic genetic framework, especially as we have access to larger data sets, which may reveal distinct clinical, social, genetic, and anatomic subtypes that we can place into models to better understand subtypes of neuropsychiatric symptoms, both in developing new treatments, but as well as testing the ones that we have, such as with pharmacogenetic research, both testing for target engagement as well as progression. And we hope that these approaches will give rise to a better understanding of these symptoms, but also better therapies. So with that, I will conclude my remarks, and thank you for your patience and attention, and be happy to engage with you rather than try and answer these questions, but engage with you on a discussion of this topic. Thank you.
Thank you, Professor Nowrangi. It was very easy to follow. And very, very deep in some points, introducing a new framework indeed because as you know, this audience combines both basically and clinical researchers, and that creates a lot of interactions between genetic anatomy and clinical output that at the end is what you treat in a daily basis. So I'm sure there's a lot of questions. I'm sure Panos and I have many questions. Can you stop sharing?
Yes.
So okay, thank you. That will help for the audience on the Zoom, but there's also audience on the YouTube. So the floor is open for questions. I'm sure Panos has many because he already told me. Pano? Yep.
I think that's a great question. And in thinking about how we think about psychiatric and even neurological diseases, may shed light on it, right? Because you have periods of relapse and remission, periods of exacerbation, periods of better health. And so many conditions all across medicine, I think, behave in this way. I think to assume that neuropsychiatric symptoms are constant, just as you said, is not doesn't map onto our clinical observation. But to think of them as exacerbating and improving symptoms over the course of time, and to map onto that what may be these psychosocial triggers that may make worsen and make better, I think would be a useful way of thinking about it as a complex mechanism. Now, I think what's helpful is that we not think of the anatomic genetic framework necessarily of taking into account all the complexity of the organism, in this case, a human being, interacting with their environment, right? There are gene and environment interactions. There are interpersonal interactions. For example, a caregiver that's upset or mean with an individual with agitation may bring out a behavioral outburst, or a stimulus from the environment where an individual may say something that's misinterpreted as a threat may emerge as sort of a paranoid delusion. So as a long way of answering the questions, I think that's exactly right. These symptoms come and go, are stimulated and recede based on the interaction of the organism with the environment.
Pano, you're muted. Yes. Any other questions? Otherwise, I could ask one. I can go. So thanks again. But what do you think about the use of antidepressants? If we believe that the monoaminergic system is contributing a lot, and I do believe it. Okay, I do believe it. I'm saying from the very beginning. Do you think there will be time frame where you can benefit more from antidepressant treatment or antipsychotic treatment in these patients? Do you think it has to do with the progression, the clinical progression, has to do with the molecular, the anatomical progression? If there is any benefit periods or periods that the patient will more benefit, because if we go back to literature and we have discussed, we have opened, this point for discussion many times in this HIAAD Bridges seminars about antidepressants, if they're helpful or not, if they can really stop some of the molecular mechanisms of amyloid beta or tau, or if they are, just, helping something that is already dying. So you put a new way of seeing the whole thing that is based also on the different polymorphisms that I or Panos has as a patients, and that can affect the effectiveness of or the help of any antidepressant or any antipsychotic treatment. But is there, besides the individual, is there any time window where you think would be better, or any scheme of clinical symptoms that are indicating more helpful treatment with antidepressants for patients or antipsychotics?
I think that's another really important clinical question, and also one of theoretical importance in terms of the lifespan and the progression of the disease. So one thing I didn't talk about that I know Costas and other people like Dr. Ismail in Canada have looked at is mild behavioral impairment. And these are the earliest presentations of behavioral symptoms, sort of in lockstep with the earliest cognitive symptoms in mild cognitive impairment. And if you reason that if we follow this disconnectionist framework, that clinical symptoms in real life, as we observe them, are a reflection of the coherence of these cognitive and behavioral networks. It would seem that, and if these networks then underlying them are the neurochemical pathways like serotonin and dopamine and so on and so forth, norepinephrine, that you would stand a much better chance of treating the symptoms if you detect them the earliest, when these networks are still fairly connected or coherent. I think that makes some sense. The more that they degrade and the more disconnection that you experience, you could also reasonably say that the transmission of the neurochemicals between along these pathways, white matter pathways or what have you, also breaks down. And so the effectiveness may also break down. So I think earlier, obviously better. That doesn't come as any surprise. But the added problem is that as the individual typically ages, the symptoms become worse in theory because the disconnection worsens. But also what we find is that there are these other co-occurring pathways and mechanisms that we don't treat now, things like neuroinflammation, immunologic response, and things of that nature. And so this is the idea with a cocktail approach, right? How do we find subgroups of individuals, let's say with psychosis, but also ones who have inflammatory markers, whether they're both we can find in the serum or in the cerebrospinal fluid or other places that we can provide a combination therapy for as the degree of disconnection progresses, that we can only hope will decrease the severity of those more severe symptoms.
Okay. And I have a second question related to the amygdala indeed. What is your view on the role of amygdala of all these neuropsychiatric symptoms? Are we having a malfunction in loss of function or disconnectivity, or you think we have a hyperactivity more similar to depression or more similar, to stress-related, network matrix dysregulation?
Your guess, Giannis, is as good as mine. In part because the literature is kind of all over the place, especially the functional MRI literature, right? And this is something we talk about all the time, is what is actually going on when we see increased or decreased connectivity? Is that better or worse in terms of more or less in terms of the presentation of the symptoms? And to what extent does compensation of one connection relate to increase or decrease in the symptoms? I don't have a good answer for that. We do know, however, that in the sort of the animal literature, this is mostly put forth by Marcel Meslim, is that widespread destruction of the amygdala creates really bad symptoms. In the macaque monkey, for example, they exhibit the Kluver-Bucy types of symptoms and things like that. So you can only assume then that without overwhelming compensation, that destruction of the amygdala or a disconnection from
disinhibition,
disinhibition from the amygdala can create worse symptoms. But it's really, these are reciprocal connections with surrounding structures within the paralimbic and limbic lobe. This issue is interesting.
The reason I'm asking and trying to connect more to the molecular base or the synaptic dysfunction or disconnection syndrome, let's say, that I do believe for many years, and I was happy to see that you're introducing more this idea, is the fact that in amygdala specifically, to more than 40 or 50% of cases, in many of the AD brains, we have an accumulation of synuclein, which is a very strange molecule that has a synaptic function. However, it's always accumulated, or in many cases accumulated specifically in amygdala. So I'm trying to see your view based on the both genetic and anatomical network, because you are really introducing a new approach, whether we could get more close to things we are seeing, but we have no explanation. You know what I mean? So the dysregulation of the network that comes and forces amygdala to overburn, if we have it, and for sure, if this malfunction is in the beginning, probably we'll see increases. You see in many literature, in many papers, and then if you go later, you will see it's burnout. So it's decreased. The activity is decreased. So how would you see whether not only the monoaminergic hypothesis or next, but also include other synaptic proteins and polymorphisms? Because synuclein is a very characteristic accumulation we have in almost 2 to 3/5 of the AD cases. And if you speak with your anatomists, they will really agree with that.
I think it's really great, and the fact that you have a deep knowledge of this outpaces my own knowledge about the synucleinopathies and things like that around the amygdala specifically. But I think that is, I think we're probably on the verge of understanding structural genes of specifically centered on structures of interest. Let's put it that way: the amygdala, temporal lobe structures, the limbic lobes, and so things like GWAS studies around the proteins that come from the genes and gene polymorphisms of those areas, I think will give a lot of information on what types of targets, pharmaceutical targets, that we can develop specifically for
exactly.
So I agree with you.
Pano, any more questions? Because overall, I think you would believe that we need. Okay, that's a dream or completely hypothesis. But do you think we should dream for more specific drugs, going far from depression drugs to treat AD patients, the neuropsychiatric symptoms of AD patients? Do you think we need more specific drugs than broader action drugs we use for depression?
Yeah, no, I think that's certainly one way of looking at it in terms of, how do we get down to specific gene polymorphisms with gene products that are, directed at finer sense of, like, serotonin receptor, for example. Or another way of thinking of it is, what are the combinations and cocktails, as I introduced, that might incorporate other adjacent mechanisms that may be affecting the function of serotonin.
Okay.
May I ask probably the last question? I am wondering if there are neuropsychiatric symptoms which could contribute to the diagnosis of Alzheimer's disease, because they are very common, but somehow we have neglected. It seems that we have neglected, or the classification systems have neglected, their potential contribution to the diagnosis of Alzheimer's disease. I am sure that not all of them could contribute or could help clinicians to establish the diagnosis of Alzheimer's disease, but there are symptoms, neuropsychiatric symptoms, which are persistent, which persist, and they are there for a very long periods of time, and they could probably contribute to the diagnosis of Alzheimer's disease.
I think that's a wonderful research question. I'm not aware of research that has established a consistent neuropsychiatric symptom across the lifespan or the progression of the disease that may have a more powerful effect on diagnosis.
Apathy, for instance, I am thinking about apathy.
Yeah, no, exactly. I mean, just taking the case N of 1 of my mother-in-law, that's how it started with her, even before we worried about cognition necessarily, that she was just much more complacent. But yeah, I agree with you. And I think that apathy may be a very strong candidate for an early warning sign, if you will, that there's impending cognitive decline. Motivational drive, right? I mean, I think that's what we really talk about, is motivational drive in anterior structures of, like, the anterior cingulate cortex, where you may or may not see the spread of amyloid into that early on. But yeah, I think that's a great concept.
Okay, I think Pano, you want to what we close together? I think that's already a great honor to have Professor Nowrangi here with us. Once more, we would like to thank you for a great presentation, introducing a completely new context or tool of thinking. This is what we need actually in the field. And hopefully different members of the HIAAD and different students, researchers, clinicians would in contact with you, developing more tools between the two parts of the Atlantic Ocean. Thank you again, and have a nice morning there. Have a good afternoon. Thanks, everybody. And we will be again together next month with another HIAAD Bridge seminar. Have a nice afternoon. Thank you.
And a good night to you.
Bye-bye.
Thank you. Goodbye. Goodbye.